día de hoy. Primero les voy a dar el aviso de seguridad. Este consiste en que en cualquier caso de emergencia, la mitad del auditorio tiene que evacuar por esta puerta y la otra mitad por la otra puerta. También les voy a pedir que las personas que estén cercanas a las puertas nos ayuden a abrirlas. Hay que abrir las dos hojas, entonces hay que quitar los seguros de arriba y de abajo para poderlas abrir. Lo siguiente es que les vamos a agradecer que no consuman ningún alimento ni bebida dentro del auditorio y que nos hagan favor a partir de este momento de apagar sus celulares. El día de hoy tenemos el placer de recibir a la doctora Teixeira-Costa, que es invitada del doctor Marcelo Pase. Luisa es estudiante de doctorado de la Universidad de Sao Paulo, en Brasil, y es becaria del Consejo Nacional de Desarrollo Científico y Tecnológico. Su línea de investigación se ha enfocado en el entendimiento de la relación hospedero-planta-parásita por medio de estudios anatómicos y comparativos. Ella hizo una licenciatura en Ciencias Biológicas en la Universidad Federal de Sao Paulo, en Brasil, y una maestría en Botánica en la Universidad de Sao Paulo, en el mismo país, y ha realizado 3 pasantías en la Universidad de Leicester, en Inglaterra, Universidad de Leeds, y en el herbario de la Universidad de Harvard, en Estados Unidos. También ha trabajado como consultor de botánica y forestación en el Instituto Butantan, en Brasil, y ha trabajado como profesor asistente, dando la clase de anatomía vegetal y de la madera en la Universidad de Sao Paulo. La producción científica de Luisa incluye artículos de investigación, capítulos de libros y apuntes cuyo tema principal es la relación hospedero-planta-parásita desde un enfoque morfológico. Entonces, adelante, Luisa.
Good morning. I apologize. Start by apologizing, but I'll be speaking in English because I feel more comfortable speaking in English rather than Spanish. Hope you all can understand me. And thank you very much for attending the seminar this morning. I'll be presenting what I've been doing in my PhD course. I'm on my very last 5 months, and this is a part of my study that I have been dedicating the last 1 year, I guess, which has to do with tomography and 3-dimensional understanding of how parasitic plants attach to their hosts. So I'd like to start by talking about what is a parasitic plant. If we go on the internet and type parasitic plants, these are all the kind of images that eventually will show up. But when I talk about parasitic plants, I'm referring to those who have this organ which we call, we call host-paraitic, which, as mentioned by Dr. Kite, embodies the very idea of plant parasitism. And the host-paraitic is an organ, just like the roots or the leaves. So it is composed of different tissues, different structures, and it acts on penetrating the host and eventually establishing the vascular or even phloem connection between the parasite and the host. So you cannot be a parasitic plant without developing a host-paraitic. And there's a great variety of parasitic plants. During the research of parasitic plants, it was thought that they would compose one singular group, but they do have a great variety. And here we have a tree, which is a parasitic plant, this tiny purple flower thing, which is a Mexican species called Lanoa. It's also a parasitic plant on the roots of neighboring plants. Castilleja, which is also a North American species. This is Castilleja mexicana, and it's also a parasitic on the roots of neighboring trees. A mistletoe, which is the most usual parasitic plant we can see, and what we call the queen of parasitic plants, which is Rafflesia. So parasitic plants comprise 1% of all living angiosperms. That's quite a lot of species. And we currently believe there has been 12 independent evolutions of parasitic plants just within the angiosperms. So there is no non-parasitic plant among the conifers. And here we have the angiosperm phylogeny, the most updated version. And you can see the green spots here, they point to the orders that contain parasitic plants. And these 12 lineages comprise 23 families, and most of them are within the order Centellades. And this huge diversity this huge morphological diversity of habits, evolutionary diversity of parasitic plants is what has caught my attention for my PhD project. So, given that the Host-Arium is a homoplastic and very diverse character, I'm trying to understand how it has evolved within each of the 12 lineages and what are the structural similarities and differences of this organ among the lineages. And that's part of what I'm doing at the Universidad Autónoma de Aguascalientes. So I'm doing a short stay here in Mexico, working with that. And when I talk about structural diversity of the Host-Arium, that's some of what I mean. So here we have, in this very short phylogeny, just the 12 evolutions of parasitic plants, and they are coded according to their functional mode of parasitism. So we have the root parasites that that are capable of performing photosynthesis. We have stem parasites, and here are grouped most of the mistletoes we know. Root oloparasites, which are parasites that attach to the root of their hosts, but they're not able to do photosynthesis, so they usually attach to the host phloem as well as the host xylem. And here we have the most weird thing in reality among parasitic plants, which are what we call endophytic parasites. They live their full lives within their host, and we can only see them. Here we have the flower buds for Pilostyles, which is a genus that also has species here in Mexico. But here we see the flower buds, and so it will be, the flower buds will be opening, and then this plant will have its flowering and fruiting. But during its vegetative phase, we can only identify them as these tiny parenchyma masses within the host bark. And despite their great diversity, of course, parasitic plants also have similarities, and that's where my research with parasitic plants has started, actually. During my master's degree, I was comparing the structure, again, the structure of the Host-Arium of two philodendron species that occur in South America. And what struck me first is that despite their morphological similarity, their anatomy is quite different. So here we have this philodendron peloteti, which makes a more localized infection through in the host. So here we have the tissues of the parasite and the wood of the host. And compared to these other species, Philodendron bathyorictum, and here we see the parasite, and it's here, and it's also here, and it's also here. So we can see that this one is a bit more dispersed within the host body. And these were the first anatomical slides I was able to prepare for these two species. and wow, they're completely different. But could it be that I sampled one segment that was different between those two, or the general structure is in fact different? So I thought, okay, I'll just cut a few more slides in other regions of the plant, okay? But they're not what we usually work with in plant anatomy. As you see, there's a hand, and all this is the whole story of the parasite. So they're huge, and they're quite difficult to work with because, first of all, it's wood, so it's not the delicate flowers that we usually slice with the rotatory microtome, and they're huge. So embedding and big materials take more time, of course. Slicing requires the sharpest of the knives, and then afterwards the knife is just almost ready to throw away. And if I attempted to slice, to produce anatomical slides from here and then here and then here and in all different directions, I would have a gigantic jigsaw puzzle to assemble in the end. Of course, it has been done. It's how I used to do anatomical research with these plants, but the ideal tool would be to use X-rays. And the super ideal tool would be to have X-ray vision, so that I could analyze these plants not only as a slice in X-ray, so I could analyze them as a whole. And that's exactly what high resolution X-ray computed tomography does. So this is a tomography scan of the species here, a tomography scan of this one another, and we can see the difference, right? These, all we see here in white is the parasite, and all we see here in whitish color is also the parasite. So as I said before, this one is much more dispersed within the host body than this one. So the high resolution X-ray computed tomography, or in short, microtomography, as we usually call, when I started working with that, my first concern was, will I be able to identify the same general structures that I'm able to identify using traditional techniques? So, and to my great surprise, to my great happiness, yes, we can see the same structures here and here. That's what we call, for this mistletoe species, cortical strands. So we see them here, we see them here in a morphological view. And depending on the size of the structure, we can even see the same structure using microtomography and conventional anatomy. So, and the great part, the fun part is that we can produce video. So all we see here changing and shifting in white are parasitic tissues, and here we see just a bit of the host wood. It will come back again so we can see it better. Oh, there it is. So all we see here in white are those cortical strands that I was talking about, and here is the main body of the parasitic plant. So what I was able to find out right away is that the microtomography techniques work very, very well for large samples. It allows a 3D observation. It's very good for comparing general patterns, and it's a non-destructive technique. So I can use that very same sample to do a specific anatomical analysis later on if I want to. So these first results we published 2 years ago in 2016, showing this case study of 2 phorhodendron species. And here we have a bit of the a bit of the non-fun part, I would say. So for this paper, we worked with 6 samples, and we can see here we have the size of the samples, and this one is 10.5 centimeters. And look, it took me 3 hours just to scan that sample. And then roughly 2 other hours to reconstruct the scans into a 3-dimensional object. And that is because, sorry, I was using this machine, which is a tomograph that is adapted to use with live beings. So you can put a live rat, as you have here. It has a little oxygen tube, and you can put live plants inside the tomography as well. If you have the plant in a little pot, you can put the live, you can put the live animal or the live living plant in here. And because it works with living specimens, the X-ray dose has to be much lower than what we would have, in a, for instance, an industrial tomography machine. So that's why it takes so long to scan the samples, because the X-ray dose is much lower. So the animal or the plant will roughly survive after the scanning. And in this first pool of results, I also worked with contrasting agents, because we could see that there was a very good natural resolution. There was a difference in density between the tissues of the parasite and the tissues of the host. But what I wanted to do was to see whether I could increase this density difference. So the first contrasting agent I worked with was lead citrate, and they had to be heavy metal salts, because a tomography machine and X-ray machine depends on the difference of densities among tissues. That's why when we do an X-ray, we don't normally wear rings or necklaces and stuff like that. So this is the apparatus, the funny apparatus we set up to inject the contrast. So here we have the host, and this little ball thing is the parasite, and we would have the contrasting agent, the solution, in one of those hospital bags, and it would be attached to one of the branches of the parasite. And the thing with this lead citrate contrasting agent is that it goes within the vessels. So all we have marked here are the vessels of the parasite, and a little bit here of the vessels of the host. And this was actually an adaptation of a very known and established technique, published originally by Dr. Sperry in 1988. In this work, Dr. Sperry used safranin as a way to differentiate functional and non-functional vessels in wood. So then I adapted that using heavy metal salts. And this is one of my favorite images of microtomography analysis. Here we have the host. Here we have one parasite and the other parasite. It is the same species that attach multiple times to the same host. And we can see that, so I was injecting the lead citrate in here, and we can see that it flows to the other region of the parasite in a much more concentrated volume than it flows towards the host. And that allows us to kind of map the set flow between the parasite and the host, which again, it's something that has been done before, but not with a 3-dimensional visualization. And our apparatus was based on the apparatus of Dr. Sperry. This is actually an image from a much more recent paper, but just to show you that it works in the same way. We have a reservatory with the solution we want to perfuse through the plant material, and it's elevated from the ground level where the plant material would be, and that height difference promotes the pressure difference that makes the liquid flow through the plants. So it allows us to, with the microtomography, to do set flow analysis and mapping. With these lead citrate contrasting agents, I was also able to improve a lot the anatomical analysis. So here we have another parasite. It's called Sebium fungiforme. It's a Palinophoraceae. And here the arrows are indicating the host root. So it's a tiny host root that is attacked by this huge potato thing, this huge turbicle of the parasite. But although it forms this huge turbicle, the actual interface area, the actual area where the parasite penetrates the host and connects its vessels with the vessels of the host, is very tiny. So this can be quite huge, but this area would still be 2 to 3 centimeters thick. So if I'm doing the sectioning and I miss this 2 to 3 centimeters thick area, then all this material is not of interest for me anymore. So what I did with microtomography, let's see. There we go. So here we have the host root, and I hopefully you could see that the solution I injected, the lead citrate, penetrated the, here it's coming back again, so it goes through the host root, and in this point it diverges to the parasite. So if I make little marks on my sample, I know exactly in what position this happened. So I can retrieve that sample after the tomography and do the precise anatomical and even electron microscopy analysis, because I will know exactly where the parasite attaches to the host, where the vessels of these two plants are connected. And so we have further results with this technique. It allows us to do set flow analysis and mapping, and it allows us to locate regions of interest and therefore to improve traditional anatomical analysis. But on that paper I also worked with another contrasting agent, Lugol solution, which is basically aqueous iodine. And as we can see here, the Lugol solution has a very different effect of the other contrasting agent, the lead citrate. So Lugol solution is usually applied to identify starch. So what we have here is that this solution does not flow nicely just within the vessels. If there is parenchyma tissue around the vessels, and this parenchymatic tissue has starch, the Lugol solution will spread through everything that has starch. So it allows me to, again, identify regions that are starch-rich. And this has proven to be very useful for what I have called the case study of endophytic parasites. And I'm going to show you a series of examples in which I have used this technique to locate endophytic parasites. So to start off, we have here Arceuthobium pusillum. It is a widespread species in North America in general, and just so you know, it is a parasite of conifers, and here we have the conifer leaves, the parasites are just these brownish things. These are the inflorescence of the parasite, and these green bobs here are the fruits of the parasite. So we can only see this plant when it's flowering or fruiting. And when we do an anatomical slice, first thing we think of is, where is the parasite? And it's very little tissue here and that as well dispersed within the host body, but it's very difficult to identify it. So using this technique of injecting Lugol solution, I was able to identify the parasite much clearer. Where is the... up there. So all we see here in white is the parasite. Here is a flowering shoot of the parasite. Here is all host wood. We can see even the growth rings of the host. And we can see how the parasite penetrates the host wood. But if I analyze a region that is very much infested with the parasite, I can roughly see that with anatomy. The thing is that we were able to locate parasitic tissue within the host even before the parasite enters its reproductive phase. So we were able to locate this parasite without knowing it was there from a morphological point of view. This is quite interesting because Arceutobia bacillum is known to be one of the Arceutobia species that actually colonizes the host apex, the shoot apex, and that's how it spreads through the host. As the host grows, the parasite grows with it because there are parasitic tissues within the host meristematic apex. So we were able to locate that and to identify, well, this tree has a parasite without seeing the parasite. A similar thing was carried out with Pilostylis blanchetti, and that's an even more, I would say, rare species in the aspect that the endophyte, which is how we call the parasitic tissue that is within the host, so the parasitic tissues are much more reduced when compared to Arceutobia, for instance, and this is one of the parasites we have no idea how they contact the host in the first place. When we see a host stem that is already bearing parasitic tissue, fine, we can understand how they connect to each other. But how did the parasite ended up here in the first place? What happens with Arceutobia is quite different. As most Mistletoes, Arceutobia has this kind of gelatin protein around the seeds in the fruit that helps the seed attach to the host branch, in the case of Mistletoes. This plant has an entirely different dispersal mechanism, so and it's very interesting because it also colonizes the host, but it seems to colonize the host from the bottom to the top. So here we have fruits of the parasite and flowers, flower buds, and here we have the apicalmost region of the host where we can rarely see parasitic tissue. And again, using the Lugol solution injection technique, we were able to locate parasitic tissue within the host bark, but without seeing the parasite on the outside. So here we kind of have a little bump here and here, but if we look here at this black arrow and here, there's no sign of the parasite outside, and we were able to detect the parasite even so. So what we're hoping to do for the next steps with Pilostylis is to try and scan other regions of the host, and even the roots, to see, to test the hypothesis that this parasite is actually able to penetrate the host by the roots, and it would be kind of hidden within the host roots, and then it would make its way up to the host stem. And that is very interesting, this topic of endophytic parasites, because endophytic parasites are present in four groups of parasites. So Citinaceae, which has representatives in Mexico, Epodontaceae, which is the family of Pilostylis, with representatives in Mexico, Rafflesiaceae, and Santalales. So I was able to work, as I just showed you, with Ursiotobium, which are here within the Santalales, and with Pilostylis. I have recently sampled Dalophytum, which is the Citinaceae, and I sampled them just last week in Morelos, and I'm eager to see how they spread to the host root, through the host root. But what I think is very, very great of this tomography analysis is that it works for these tiny, tiny parasites, as well as it works for the plants that produce the largest flowers in the world. So I was able to work with Rafflesiaceae species, and we can see here that their distribution is very restricted to this region of the world. Then, there we go. They produce the world's largest flowers, and it's not an inflorescence as Morphophyllum is, it's just one singular flower, but they have the most reduced endophyte. So it showed that it was hard to find Ursiotobium within the host wood. It was a bit harder to find Pilostylis tissue within the host. This is nearly impossible to find. Before the formation of the flowering bud, these plants behave, or these plants can be seen as individual parenchyma cells within the host. And their hosts are vines, are lianas, which usually have loads of parenchyma tissue. So you're looking for one parenchyma cell within a bunch of host parenchyma cells. And that's one reason why our current understanding of the life cycle of these plants is not complete. We have restricted comprehension of this life cycle. Here we can see how it starts. Look, there's 1, 2, 3 parasitic cells within the host tissues, and these parasitic cells start to divide, and then we have the flower bud. Once we have the flower bud, things are much, much easier because we can locate actually the parasite. But what I was able to do is that, so here I injected also a gall solution. Everything we're going to see in white is the gall solution that flowed through the host tissues, and then here in black we can see that there's huge spaces in black. This is parasitic tissue because the host will have starch in its parasitic cells, but the parasite will not. So here we have the largest piece of parasitic tissue, but it's also here. So that is a way we can locate even not solitary cells, but up to 5 groups of 5 cells within the host root using this technique. And of course, I'm hoping, let's see that again. I'm of course hoping we can use this technique to also try and understand better how the life cycle of these plants work. So again, here we see the parasite, It's quite different from the opening of the host vessels because the parasitic tissue takes a larger space, right? So my take-home message is that I guess I said enough how great is microtomography analysis, or at least how great I think microtomography analysis is, but it is possible to do all this kind of stuff that I've been showing you, even the understanding of the life cycle of plant species. All this is possible to accomplish with other plant species, and it has been done. So the literature in microtomography analysis is fast growing, and for instance, there's regarding life cycles, there are people working with flowers and fruits of plants doing tomography analysis to see how they develop, locating. There's a lot of work that has been done with wood, trying to locate particular regions of fragility within the wood, especially wood that is used for commercial purposes and stuff like that. There's a very interesting group working with set flux using tomography analysis. So it's a very, very powerful tool that it's becoming more and more available in different universities and research institutes around the globe, I guess. So I would like to thank you all for coming, and I would like to thank the institutions that have welcomed me, my supervisors in Brazil, in Mexico, and in Harvard, the institutions that gave me the scholarships for do all this fantastic tomography work, and of course the UNAM for inviting me for this seminary. Thank you very much.
Muy bien, tenemos suficiente tiempo para varias preguntas.
Thank you for your very interesting talk.
Thank you.
It brings, it is a surprise to me that there are no ferns or monocots.
There are parasites, yes.
Yeah, is that a consequence of not having wood?
No, I don't think so. There are ferns and monocots that serve as host species. Actually, the family Urbancaceae, for instance, is widely known for parasiting monocots. But we quite don't understand why the parasitic evolution has been restricted to the Angisperms and the Eudicots. But it is possible that there was a parasite among these other groups, but it's now extinct or something of this sort. But they do act as hosts, not just as parasites.
¿Alguna otra pregunta? Sí.
Do you know if all these parasite plants are dispersed by animals?
And some of them are, some of them are not. The thing with parasitic plants is that nearly any question you ask, the answer will be it depends, because it has this great variety. So most mistletoes are bird dispersed, but there's a lot of plants that are wind dispersed, which is usually a problem, because for instance the most severe agricultural pests that are parasitic plants are wind dispersed. So if you're trying to contain the parasite and forbid it to spread to the other fields where you're planting maize, for instance, it's probably not going to work, because the wind is going to carry out those tiny, tiny, almost orchid-like seeds to everywhere else. Rafflesiaceae, we believe it is dispersed by mammals. There has been some cases of mammal dispersing parasitic plants, but it's mostly wind and birds for mistletoes.
Yeah, because I noticed that several of your families have fruits are dispersed by animals, like carnaria.
Yes,
And it seems that it's normal that animals not only disperse the thing, but they plant the thing.
yes, yes. What happens with, so mistletoes are the most, the parasitic group that we understand best how the dispersion works. So the bird will eat the fruit, and when it's trying to remove the seed with that sticky substance from its beak, it will place the seed in the host. With the other species, we have this gas, especially the mammal dispersed species, we have this gas that they're able to bury the seed of the host and then, the seed of the parasite, and then the parasite will germinate on the ground and find its host. It has been shown for, I guess, the parasites within Aristolochiaceae and one or two species within Balanophoracea, but we have no proof, no evidence of this being the case for Rafflesiaceae or Pilostyles or this more reduced and hard to work with parasitic plants.
Thank you.
¿Alguna otra pregunta? I have a question. Do you have similar information for every diverge lineages within Angisperms, such as Ignora? And also, do you tell us about the life cycle of Ignora?
Ignora is very weird as well. They have been actually termed the most weird plants in the world. And what we know for, I guess, one or two, as well with Ignora species, is that they are mammal dispersed, the seeds are mammal dispersed, and then the mammal would bury the seed in the ground, and when the parasite germinates, it finds its host. So, but it will be, most of its tissues will be underground, and then we only see the parasite as well when it flowers and when it's fruiting, but they do live outside the host branch, or actually the host roots. So when we dig to sample material for the Haustorium material of Ignora, we actually find like rhizome-like structures of the parasite, so it's quite dispersed, and it's able to attach to the host in several points, usually. But they are very under-researched, I would say, because they're hard to find, and they're hard to sample, because their global distribution is restricted to, so I guess we have 3 or 4 localities in Latin America, and all other species are in African countries, which are usually at war. So it's quite tricky to sample them. I was able to sample one of them in Brazil, but it was already dying due to the dry season. But they are very, very nice when you are able to find them, and they're very poorly researched.
Well, I didn't know they were also distributed in Latin America. So was the similar life cycle for Prosopontes?
Yes, yes. It's so we have Prosoponte in Africa, and we have 2 Ignora species in Brazil, for instance. 2 or 1, there's a debate whether there are 2 different species or 1, but their localities in Brazil are down in the southmost region of Brazil and up in the northmost region of Brazil. So we believe there could be a continuum in this distribution, but because they're so hard to find, We actually don't know.
Thank you very much. ¿Alguna otra pregunta? Bueno, pues les agradecemos mucho su asistencia y tenemos ahora tiempo de platicar con la doctora Luisa en el momento del café. Muchas gracias.